Hello, and welcome back to another exciting episode of What is Biology? I'm your host, Mr. Johnny Hopkins, and today we're going to be talking about genetic vocabulary, just a little hodgepodge of some random stuff like last time, and we're going to just keep learning a little more about genetics and some weird stuff like that, and maybe something will come out. So, today we're going to be covering just kind of general stuff about integration, like with vectors and plasmids and bacteria, and kind of like the basics of expressing a protein inside a cell. And then some experiments you can do on like getting the foundations behind going and trying to start expressing a protein inside a cell. So, first of all, gotta cover this thing called recombinant DNA. Recombinant DNA is just DNA that is made from a non-native source. So it's essentially the DNA that you make in the lab that is made in a test tube, whereas compared to this guy right here, that DNA, you can see that DNA, right? That DNA is a native one. It is not the recombinant DNA. It's made by the native source. It's the recombinant or whatever, the native, you know what I mean. So and then we have something called a vector. And the vector is just DNA that replicates within the host cell. So it's essentially what you inject into the cell. It is that guy. And so the way we typically integrate a vector is, so we have some sort of DNA sequence that like, hey, this guy is like ACCAG, CCTTACT. And you're like, man, that seems like it's gonna make an amazing protein. That's just gonna like save cancer and, you know, make me a superhuman that can shoot webs and also know how to communicate in a healthy manner with both political sides and bring them all together. Like, this is going to be that protein. So we want to integrate it into this sort of DNA and see if it actually does all those major things by testing it in some lab environment somehow magically. And then the way we want to do that is we take that DNA sequence of, where I said A, C, C, whatever, and then we have some other DNA segment that we know is expressed inside a bacteria, like this guy over here. And we take it and we use something called a restriction enzyme. And that restriction enzyme cuts a little fragment of this DNA, and then we are left with two sticky ends, this guy right here and this guy right here. And so what happens is then DNA that we want to create, that we want to insert, we also slash it up, and then we add it on here. That has A, C, G, D, and then it has whatever the fragment was, A, C, C, G, do. And so it has this, and then that's the fragment that we are expressing right there. So we cut up some sort of pre-rendered DNA with the restriction enzyme, then insert our sequence in, and then DNA ligases will attach these guys and close it up, so we have now a some sort of vector that is inside a DNA fragment that is actually expressed inside a cell. And so one thing that is typically noticed that restriction enzymes only cut at short palindromic sequences. And what the heck is a short palindromic sequence? Essentially, it's when you read it from point A from the start to the end, and then you reverse it. That's what the reverse tail is. So the 3' is the same as the 3', the 5' is the same as the 5', and of these DNA. So like you kind of just invert it, like, oh, it's the same thing. But it doesn't necessarily need to end at be like ACA, because that would actually not work. But so you think you understand what I mean. If you don't, leave comments. It'll be like the first comment that's ever left on my entire channel. So, you got this. Plasmid. So, what is a plasmid? Plasmids are typically what the vectors are. And plasmids are just small circular DNA fragments that are separate from the chromosome. So, inside a typical bacterial cell, you have the main DNA, just this big, giant circular glob. They have little tiny DNAs right over here and here, maybe one right over here. There's just a bunch of these little guys, these plasmids. They're just kind of like wiggling about, doing their thing, and having proteins be made from them. And they're usually around 2,000 to 3,000 bases long, so 2 to 3 kilobases. And then they usually are started out replication at the ORI, the origin of replication initiation. sequence, which is around 50 to 100 base pairs. That's just a little thing that you should probably be able to say at random different intervals when you are talking to random people. So, and then you can inject like plasmids and other DNA sequences into different cells. And there is a different term for what type of cell you use. It's called transformation, when you insert it into a bacteria, so essentially something that's not eukaryotic, and it's transfection when you insert this plasmid or some sort of DNA into a eukaryote, such as a yeast. That's the typical one that you express proteins with, but you can also do it with other cells, HeLa and HEK cells, most notably. And so when you, this process is typically done in an interesting way, where you have essentially you have the plasmids, and then you have your cell. This is a bacterial cell. And so you have your plasmids in your cell, and you just wait some time, you just kind of like mix them about, like stir in your pot like an old witch. The plasmids just don't enter the cell. Like, what's going on here? But then what they actually have to do inside an actual experiment is you have to also do some heat shock, where you kind of like just zap it with heat really quickly. And then you mix it around like an old lady or at her cauldron, screaming the name of Macbeth and telling them the force is going to move, is that actually the force of these mRNA are actually going into the cell, and then they actually become expressed inside that cell, and you can actually watch them create this protein that's going to solve world hunger somehow. And so you got this cell that has now these plasmids that are injected into it using using transformation, and then this cell will continuously reproduce and reproduce and reproduce, and then every single offspring from it, you get multiple offspring. And those guys would be called clones because they're the same guy as the original parent. And then we go, and here's something called complementary DNA, cDNA. So usually what happens is that if you want to analyze mRNA that's made inside a cell or something, we need to extract the mRNA, but mRNA is a very, very fragile. So what happens usually is that you take the mRNA and take a reverse transcriptase, and then you throw the reverse transcriptase onto the mRNA, and it'll synthesize DNA, cDNA, complementary DNA, and then you can analyze that complementary DNA in however you want to do. You can also observe, and that's usually occurs also with some hybridization of the cDNA ssRNA structure. So you see one chain be DNA, one chain be RNA. And so with that, that's a little bit about some vocabulary, and then we're going to talk about some very simple experiments that you can run that are, well, I mean, these are experiments, but ones that you just like instantly know when you go inside, when you look at it inside a paper. So first one is electrophoresis. This is pretty much the exact same thing as protein gel electrophoresis, but except instead this is agarose-gel. Big difference, huge difference. The idea is the same. You have a electric gradient where you send an electromagnetic pulse where one part is positively electrically charged and the other is negatively, and then the sample will go down. And if we recall really quick, DNA and RNA always have a negative phosphate group per residue, so it's always got a positive charge, so it will always migrate towards the, it has a negative charge, so it will gravitate towards the positive charge. And then we we load up a ladder, just a sample of known base pair length. Then we take our sample, we inject it, and then we can measure over time the movement, and then we can see approximately how the length of the sample is. And what we see here is essentially this is 100 base pairs, 200, 300. This guy's about 350 base pairs, our sample. And so one thing to note really quickly as well is that you can't really see this when you actually look at it, just on the dot, is that you actually have to mix it in with lithium bromide, and this lithium bromide, you need to be very careful with it, otherwise it will stain your fingers, is that it actually injects between the bases, and so it actually allows it to fluoresce very, very strongly, so you can get very strong fluorescent images, which if you remember anything about fluorescence, might shine in, might shine out. But it's on a short time scale, so And then we have something called PCR. So essentially this is a step right before this. Switch a few words. So PCR stands for polymerase chain reaction. And polymerase chain reaction is essentially you take some DNA, AP, so DNA polymerase, take some DNTPs, and you take a primer. So a primer is just like a little section that's RNA on it, and then you just increase the temperature. So you increase the temperature, that causes heating of melting of the chains, and so then DNA will associate with them with the primers, and then it will go along and synthesize another strand of DNA, and then continuously do this, and then you can just kind of have a bunch of different DNA sequences. Yeah, you can have just a bunch of the same DNA constantly, and then you can use those DNA and do some electrophoresis to make sure that it got the DNA it actually wants, and that it actually worked. And then you can do some transformation and get it to express in the cell. And then one other thing that is also important to do is something called sequencing. So we can also sequence this guy if we're also questioning some stuff over here. And so there's a lot of different techniques for sequencing, and there's a lot of really fancy ones, but we're going to just focus on the very simple one that is very textbook. It's the Sanger method of sequencing. So essentially it's just synthesizing DNA by truncation. What does that mean is that PCR, it's just very interesting to use PCR, but instead of just having DNTP, so the DNTP is like ATP, CTP, GTP, and TTP, yes. And so rather we have DDNTP, so we have an experiment with just A. Then we have an experiment with C as a DDNTP. And then we have one as T as a DDNTP, and so on and so forth. And we have each of them so that we prematurely end at an A or prematurely end at a C. One thing to note, what does DDNTP? So DNTP is a deoxynucleotide and triphosphate. And then DDNTP is a dideoxynucleotide. So if you remember, the 3' end of the sugar is what continues the motion of chain elongation for DNA and RNA. And so if we cut off that OH group right there, that means the chain prematurely ends. And so when we have it specifically just an A, that means we end at any single A sequence. And then if we do it on C, we end at that C sequence. And so if we just do that, and we end up doing electrophoresis, we get a bunch of different stripes at the regions that the A's and T's and C's and G's are, because that's where they end, where we prematurely end the DNA sequence at. And so then from there we can go and we can just look at that diagram, this electrophoresis, and we can be like, oh, the first one is T, the next one is A, the next one is actually G, then it's C, then it's T, then it's G, then it's C. We just look at this and we can reconstruct the actual sequence by itself, which is pretty interesting. But one thing to note, this only works on very short sequences. So short sequences being around 500 nucleotides. So it's not a very common technique these days, but it's the one that laid the foundations for everything else that came after. So with that, that is the most of genetic vocabulary. We have recombinant DNA, we have vectors that we insert into plasmids using restriction enzymes, and then we transform them into bacteria, and bacteria make clones of themselves. So we can also check all this stuff DNA-wise by making the DNA using PCR, putting it into electrophoresis, and making sure that we have the length of it, and then we can use sequencing on that using the Sanger method to try to determine what the sequence is by truncating it with DDNTPs. So with that, I'll end here for the day, and I will see you next time where we'll talk about more genetic stuff. Thank you.